Greetings, class, and welcome back to the last section of our unit on the cell cycle. In this short section, we're going to talk about how the cell cycle is actually regulated, how the cell knows to move from one stage to the other. And it's hard to believe that we know so much about cells and biology that this particular part of the process wasn't actually even discovered until about the 1980s. When scientists made this discovery, they recognized that there are actually special types of proteins that actually help move the cell along through the process of the cell cycle. And this group of proteins they gave a collective name to, they call them cyclins, because they're responsible for controlling the cell cycle. And basically what these proteins do is they serve as checkpoints, and they create chemical signals that tell the cell to move from here to there and so on and so forth. So for example, at the G1 checkpoint, the gap 1 phase, if no signal is given, the cell won't leave that stage and move into synthesis. It'll enter what's called the G0 phase, and it will stay there. And some cells are like that. For example, brain cells and nerve cells, some of these cells will stay like that. That's what makes them so hard to repair when they get damaged, because they don't want to move along the cell cycle. Okay, so what we want to talk about in these next couple slides is how cyclins work. So let's move on into the next slide. Okay. First of all, how were these cyclins actually even discovered? What was the scientific process? As scientists started to identify these particular substances, what they were able to do in the lab was actually take them, like for example, take a sample of cytoplasm from a cell that was in a particular phase of the cell cycle, and then put it into another cell that was in a different phase, like for example, put it into this cell, which was an interphase, and actually kick the cell into entering mitosis. So scientists have been working very hard on identifying these compounds because, I mean, their medicinal benefits could be, of course, very obvious. Maybe in the future they can be used to help grow cells again that don't want to necessarily grow, like, say, for example, spinal cord injuries or brain tissue injuries, or maybe help in dealing with cancers, which we're going to talk about in another type of slide. But these cyclins or chemical proteins can be broken up into two groups. You have internal regulators and external regulators. Internal regulators are those types of cyclins or proteins that are responsible for moving the cell through the normal process of the cell cycle, going from G1 to S to G2, moving through the mitotic process. And that's why it's called an internal regulator, because it's coming from within. Then you have also external factors. Those external factors are, like the name says, those are factors, or external regulators, excuse me, I think I said factors. External regulators are affected by factors external to the cell, and they set off triggers to let the cells need to do extraordinary things. Like, for example, let's say you get a cut on your arm. You can't wait for the cells to reproduce through their normal mitotic process. It would take too long. The external regulators will regulate that, check that, and let the cells know that they need to grow faster. It's time to start growing again and reproducing until they grow back and heal the wound. And how do they know when to stop? You know, when these cells are very smart. When they sense the contact of other cells and stuff, they can actually shut themselves down and stop the process of division because now you've healed. Now, when you're talking about cyclins and you're talking about the cell cycle, there's also another type of process we need to talk about. That's called apoptosis. Apoptosis is the controlled death of a cell. Imagine, for example, your cells never died. They stayed on forever. You would be buried in cells. That's not any good either. I mean, things would grow together, like your hands, your fingers, things like that. Cells need to know that they need to die to make room for other cells. So this programmed death of a cell is called apoptosis, and it's a normal process. Again, so it's called apoptosis, the programmed death of a cell. But what happens when you have cells that grow uncontrollably and they're not responding to the effect of cyclins? Those are called. So let's look at this last slide together as we talk about cancer. And again, what is cancer? I know you remember from the reading. Cancer is the uncontrolled growth of cells. And these cancer cells are not at all following the internal or external regulators that are triggering them to try to get them to control their growth. They're dividing and dividing and dividing, and almost like immortal. They're not responding to anything. And that can be very dangerous. Let's look at the progression of some type of skin cancer here. You can see here it begins with the genetic mutation. Perhaps for years you've been ignoring wearing that suntan lotion. You've been out in the sun, getting that UV radiation. And over time, it can cause a genetic mutation. It did for me on my face next to my nose. I had skin cancer removed there a few years ago. So that radiation over time could cause the DNA to make a little mistake when it's copying. Those types of environmental factors that cause genetic change are called mutagens. I'll write that down for you, mutagens. So let's say you have now a genetic mutation. That DNA has been changed and altered a little bit. And now those daughter cells are going to be changed and altered as well. And perhaps that mutation has caused the cell now to ignore those cyclins that would tell it to grow normally. What starts to happen is you start to get a growth here, hyperplasia. And then that growth can become more serious, dysplasia here. And it's getting more rooted into the tissues of that particular area. And for me, in my case, I started to get a growth on the side of my nose that didn't go away. So I had to go to the skin doctor to have it investigated. And they took a little sample and sent it to the lab, to the pathology lab, and they found out that it was a cancer, and so they had to remove it. And they wanted to remove it before it got too embedded into the tissue. As you can see here, the cancer can get quite embedded into the surrounding tissue. And in my case, so they had to surgically remove it not once, but twice. They go down deeper to make sure they got all the affected tissues in the region around my nose. So they went down pretty deep, and it was pretty painful afterwards, but they had to make sure they got all the surrounding cancer cells as well. And had they not, it could have gotten even more serious. And what's become an invasive cancer, like some of these types of malignant skin cancers, where they can spread to other surrounding tissues and then spread into the rest of the body, which can then be very, very dangerous. So make sure you read this last section. It's very important. And not only does it talk about regulation of the cell cycle, it also talks about when the cell cycle is not regulated. It talks about cancer and also the treatments for cancers, radiation therapy, chemotherapy, these kind of things. So that concludes our unit. Hope to see you Monday morning at 7:30 for our review for the test. Thank you.